Good morning, children. Today's class, we are learning what is this sex-linked inheritance, especially X. X means, you can see that in female, double X is present, X and X, that is in the last chromosome, that is the 23rd pair. And whereas in male, you have seen it is X and Y. So here, sex-linked inheritance means it is the appearance of a trait. The character, alternative form is called as trait. The appearance of this trait which is due to the presence of the X chromosome or on the Y chromosome. Why it is mentioned it is on X chromosome or Y chromosome? Because X chromosome and Y chromosome, they are the last pair of chromosome which is linked to the sex of an organism, which determine the male or female. So now in today's session we are learning about X-linked, means the inheritance or the character which is passing from generation to generation, which is based on this X chromosome. So you know, again once more I am telling, in female the chromosomes are XX, that is the 23rd pair, the last pair, the two chromosomes are XX in female, represented by round symbol, whereas in male it is XY and square symbol. All these things you have studied. So now you can see some genetically based diseases are there. One is called as hemophilia, another one is called as sickle cell anemia. These diseases, if it is present in any one of the parents, maybe in the mother or in the father, you can see that will be transmitted to the children. That is why these diseases are called as what? Hereditary diseases. So first we can study about the hereditary disease, that is color blindness. Yesterday I told you what is meant by color blindness. Yes, that those people who are those who are having this color blindness, they are not able to identify the colors, that is between red and green. They cannot identify it clearly. That is what is called as color blindness. So this, suppose any one of the parent is having this color blindness, that will definitely it will pass to the children. So let us see how it is passing. So here color blindness, the father and mother, see that X-linked inheritance we are studying. So that if they are a carrier or having near to the X, we are putting a 0 like this. Okay, so it is related to the X chromosome, and if a small 0 is near to this, that means either the parent is a carrier or it's a, you can see that the person is having that diseases. So in the case of a female, listen properly, in the case of a female, XX, in that near to that 1 0 is putting means, you can see that this female is a carrier, whereas she will not show it phenotypically. She will not show that character externally. But she is a carrier. She can pass this to the next generation. Whereas in male, let us see, I already told you it is XY and near the X is 0, because here she is a carrier, whereas a male, if he has this 0, you can see definitely he is a disease 1, that is he is having that color blindness, because only one X is present and that X is affected. That's why it is called X-linked inheritance. Diseases which is passed through the X chromosome. Here in this female, suppose here 2 zeros in both the X, if it is we put like this, means she has that disease and she can also transmit it. But whereas 1 0 is there, means she is having that disease, but externally she will not show any symptoms and she will not be affected by this color blindness, whereas she can pass these diseases from her generation to next generation. Got it? Here, whereas in male, only one X is present. Only one X is present, and that is affected means definitely he will be having what? Color blindness. Now, certain examples we can see. Based on this, here there is a normal mother. Normal mother, what will be the genotype? Yes, XX. Normal mother, this chromosome, that is XX. And you can see a color blind father. Color blind father, this is the female with round mark into a color blind father. XY. Okay. Here I put a small zero. Listen properly. That means he is a color blind. This is a normal mother. Normal. His father is a, you can see that he is shown with the square. This is color blind. So mother is normal, father is color blind. So what is the possibility of forming gametes here? Here there is only one possibility, that is X. Another X, that is normal X only. At the time of gamete formation, you can see, or the X formation. Here during the process of sperm formation, you can see that what is the possibility? Here one X like this and another Y like this. Clear to all of you? Here there is only one possibility, the normal X chromosome, whereas here X with a defective gene and Y normal one. So you can see while children are produced, we can show it here. So here you can represent the male and here female. Okay, this one male, what are this X, 0, Y. Here female design X, X. And so here you can do first these two. What is the possibility of their children? Here with a normal X and another X with 0. And here these two. This X with this Y. X, Y. Now coming to this corner. This X with this X. So X is a carrier and here X, Y. Got it? Once more. This X, this is from this what I wrote on the top, that is male. This is female. So here X and this X in this column, X, the second X is a carrier. Here this X and this Y, X, Y. Here this one, this two in this column, X. Here this X is a here. So here it is a carrier. And here XY, here you can see. So can you tell that this is the genetic constitution? So first one, whether it is a male child or a female child? Yes, you can see XX, so this is a female child. And this one, X1 is a male. Here XX is a female, whereas this one, last one, yes, it is because it carries XY, it's a male child. Now you can see that here this one XX is a carrier, a female, she is a carrier, and here the male is a normal boy child, and here again there is another possibility, this child will be a carrier, this is a female carrier, and whereas this male a normal. So you see here the mother is the normal one, whereas the father is a color blind. And what is the possibility? How the children, you can see that for the colorblind father, the two sons formed. Here this is a son, this is a son. Both the sons are normal. Okay? For a colorblind father, the sons both are normal, whereas both daughters, you can see that they are carriers. Phenotypically or externally they will not show this character, but still they can pass this character from one generation to next generation. These children, when they got married and when they produce their children, they may be colorblind. Got it? Okay. Now another example with the same thing we can see. One we can see carrier mother with normal father. Carrier mother, how we will show? X, X, carrier mother. Normal father, X, Y. Okay, this is female, this is male. Otherwise you will show the symbol like this. For female, round symbol like this. For male, square symbol like this. Okay, so here what about the female? You can see the mother is a carrier, but she will not show any such character externally or outside, whereas the father is a normal one. Got it? Now, how the offspring are produced? Here there is two possibility. One, X gametes or X form, you can see. One is X and the other one with X0. Here you can see that second one is with the X0. That means this X carries the gene for the disease that is causing what? Color blindness. Here you can see the father, normal X and normal Y. Understood? Now let us see what is the possibility. So this is male, here it is represented the female. Here you can see that male X, normal Y. Here X, X0. Now this X and X, you can see that here XX, this X and this Y, XY, this X and this X, X0, this X0, Y. Got it? Once more. This X with this X, XX. Okay? Then this X, this is a female egg, this is the male sperm, X with Y, XY. And this one, X0, that is a carrier one, with this X, both you can see X, X0. The last one, this X0 and this Y, X0Y. So normally what is the first child? Yes, this is a girl baby. So whether she is a normal one? Yes, this girl baby is a normal one. She have no problem, no color blindness, nothing. Go to the second one, XY. So this is a boy baby and XY. This baby also have no problem, no color blindness, nothing, and they are not carriers also. Coming to the third one, you can see X0. You can see what is the sex of this child? Yes, it is female. And she is female and she is a carrier. She is a carrier. That means externally she is not showing, but she has the capacity to transmit this color blindness from her generation to the next generation. Okay? Coming to the fourth one, you see X0Y. This is a male baby, and what is his condition? Yes, this is a color blind boy, totally color blind. Because only one X is present, this disease is linked to the X chromosome, that is X-linked inheritance. So as this is X-linked inheritance, you can see there is no more X is present here. So this boy is what? Color blinded boy. See here, the father is a normal one. Understood? The father is a normal one. Only mother is a carrier. See the carrier one, how she is passing to her the next generation. You can see she is a carrier of this color blindness, and she will carry, externally she is not showing any such thing, but you can see that she will pass this color blindness to the next generation. See, a son was born to her, and the son is having what disease? Color blindness, but the father is normal one. So understood the difference. So the first case, totally different thing we have seen. Now in the second case, you can see that the father is a normal one, but the one male baby, what is produced is colorblind one. So third case, you can see that colorblind man and carrier woman. Carrier female into so carrier woman, you can see that how it will be X10. That is, she is a carrier only. A colorblind man, you can see how it will be X0Y. You can see that carrier woman, you can see one X only is affected with the color blindness, whereas a colorblind man automatically you know the X is affected. And what is the type of X she will produce? One is X, another one with this X0, that is, she is in her egg, you can see that this 0 is present. Whereas in the male, you can see that X0 and normal Y. And how the children will be produced, let us see. This is male, this is female. So here, male, you can see X0. Y. Here X0. Got it? So these are the X, these are the sperm. Okay? So X, this X combines with this X. So here X0. Here this X with this Y, XY. And this XX, both. You can see X0, see here, 0 is here also it is there, 0, and X0Y. See this one clearly, first one X and X0, X1, X0, and X0Y. Here you can see that, what about the first child? She is a female one, a daughter is produced, and what is her condition? You can see that she is a carrier one. Externally, she is not showing any such characters, whereas her gene carries this color blindness gene. Go to the next one, XY. This is a male child, and can you say whether this child, male child, is having this color blindness or not? No. Automatically you can see by looking into this one itself, you can immediately understand this child is not having that disease, color blindness. Go to the third case, you can see this is the first time you are getting such a child. So X0, this is a rare case. This is a rare case. this is a female child. She is a colorblind one. The third one, she is a colorblind one. And goes to the fourth one. This is a male child and he is also a colorblind one. Understood? There one son is born. There is a possibility the son who was born is not having color blindness. A female child, there is a possibility she may be a carrier, whereas still there is possibility the female child, she is having color blindness, and the male child also having color blindness. So got the idea how these genes are passing from one generation to another generation. Sometimes both the parents will not show any such character, but still the children may show. Sometimes they will show either the father or the mother will show some characters, but children one or the other will be affected. So 3 cases I have shown you. In 3 cases also you understood how this color blindness, which is a hereditary disease, is passing from parents to the offspring. So there is every possibility that if the parents are having this color blindness, it will be passing from generation after generation. That's why you can see now it is not advised the marriage between the close relatives, because if they have any genetic disorder, definitely their children will get that disorder and again it will be passing to the next generation. So we have to not allow these close relatives to get married. Okay. In the same case you can see that hemophilia otherwise called as what? Bleeder's disease. What is meant by bleeder's disease? Hemophilia also, it is just, that is also an X-linked inheritance that is passing through the sex chromosome. And let us see hemophilia. This with capital H, this is a carrier. Okay. And the male, female, you can see this is X with the hemophilic person, and this XY, the same thing. You can see that X0, like the same how I have shown. Suppose this female, this is the female, this is the male having hemophilia. Hemophilia disease means suppose any small wound happens in your body. You can see that blood normally in our body within 2-3 minutes blood will clots there. Whereas in a hemophilic person you can see that blood will be oozing out continuously. Sometimes if it is a severe case means you can see that person may die by the continuous loss of blood from the body. So you can see here also hemophilia. Here a normal mother with a hemophilic father. This is a normal mother. This is a hemophilic father. So there is, just like I have shown you how color blindness. In the same case here XX. Here X0Y. So here you can see that this is male, this is female, this male X0Y, here XX. Okay. So these are the male sperms, it may carry X0 and Y, whereas the female XX only. So here you can see that X0. So come on, tell me here XY. So the third one X0 and the fourth one XY. Now come on, tell me here this is a female child, this is a male child, here again a female child, and this is a male child. And here both cases you can see the first and third case, both are female and both are carriers. They won't show this hemophilic character externally, still their genes contain this disease affected genes are present in these females. Whereas look into the boys, this male child, 2 children, both are normal, no problem for them. So, whereas here you see the mother is a normal one. Mother is not a carrier and she is not showing that externally. Phenotypically is also not showing. But still you can see that the girl babies are affected with this hemophilia. But though the father is hemophilic one, the 2 boys are not, or the male babies are not affected by this one. Got it? Okay. Now let us see. Like that with a normal father and a carrier female also, how I have shown you in the other case, color blindness case, how it happened. The same thing only following here. Now let us see yesterday I have told you about Gregor John Mendel. Who is he? He is the father of genetics. Why he is called the father of genetics? Because he has done a lot of studies and how these characters are passing from generation to generation. He was an Austrian monk, and in his garden he conducted so many experiments. And even now also the same principles are only in the modern science also. The same principles only we are following. So he is called the father of genetics. In short form we will call him Mendel. M-E-N-D-E-L, Mendel. He is called the father of genetics. And he took a plant called garden pea. Pea plant, all of you know. The vegetable pea. Peas, you like to eat it. That plant only he took for his experiment. There is some reasons why he selected this green pea or pea plant for his experiment. Because this pea plant have first thing, it has got a short life period. That is, lifespan is short. Within 3 months, if you put a seed of this green pea plant in the soil, you can see that within 3 months it will complete its life cycle. It will germinate, it will grow, it will flower and it will die. You can see within 3 months, because a short time only is needed for experiment. Suppose a plant, if it takes 1 year to grow and bloom, to produce flowers, it will be difficult. We have to wait till 1 year to complete our experiment. Then for the next generation another 1 year. So here pea plant, you can see that it has a short lifespan, and many varieties are, second reason is that many varieties are available. You can see in the garden pea plant many varieties are available, and you can see true forms we are getting, not the mixed form, true forms we are getting. And another reason is that they can be pollinated artificially. Usually in pea plant self-pollination takes place, but we can do artificial pollination also. How we can do artificial pollination? Yes, either by cutting the female bunch or by cutting the male bunch and then covering it within a polythene bag. So we can prevent self-pollination. Easily we can carry bunch, cross-pollination. So these are the reasons why Mendel selected this garden pea plant for his experiments. And you can see there are 7 contrasting characters also he noticed in green pea plant or garden pea plant. He noticed 7 contrasting characters. Last class we have seen. Can you tell one example of contrasting characters? Yes, last class you have seen in human beings about the nature of the hand, right-handedness and left-handedness. 2 contrasting characters, lips thick or thin, eyebrows, you can thick and bushy, then very thin eyebrows, color of the eye. All these contrasting characters you have seen. Like that, in pea plant, there are 7 contrasting characters are there. Which are those contrasting characters? First one is color. So, you can see he has taken 2 examples. In pea plant, you can see purple color is there for the flower and white color is also there for the flower. So, purple colored flower is there and white colored flower. 2 contrasting characters of the flower color. Next, come to the seed color. Seed will be formed in that, again 2 contrasting characters are there. One is yellow and the next one is green in color. So, yellow colored seed plus and green colored seed. And seed shape. Usually you can say that the seed will be round in shape. So, here in pea plant, 2 contrasting features are there. One is round and another one is ringled like this. So, wrinkles are there on the seed. That is the third one. And the next one is pod color, that is green and yellow. Pod color means you can see that green piece that like this it will be there, and inside that these seeds will be like this, isn't it? This structure that is covering that, this structure is called as what? Pod of the seed. So this pod, 2 contrasting characters is there. One is green and the other one is yellow. And coming to the next pod shape also, you can see one is inflated and the other one is constricted. So inflated means you can see like this. Constricted means like this it will be there. So pod shape also 2 it will be there. It will be inflated or constricted. So next one, the flower position. You can see that position of the flower. There are also 2 contrasting. One is axillary and the other one is terminal. You can see that axillary and terminal. Next, plant height. Height of the plant. Tall plant is forming as well as dwarf, short, the tall and dwarf, short plant, tall plant, dwarf plant. So these are the basic 7 characters this Mendel who observed in this pea plant. So once more, what are those 7 characters? Flower color, it may be purple or white. Seed color, it may be yellow or green. And seed shape, round or wrinkled. Then pod color, green or yellow. Pod shape may be inflated or constricted. And flower position, axillary or terminal. Axillary means from the base of the leaf it is starting. This is the stem and this is the leaf. Axillary means from here the flower comes. Okay, terminal means at the end region flower comes, it is called as terminal, axillary or terminal. And again, plant height, it is tall or dwarf. Dwarf means short. In all these cases, you have seen purple, white, yellow, green, all these cases, you can see that dominant one is there and recessive one is there. Dominant means super ruling one. And next one, you can see that recessive means in presence of dominant, it will not show that character. That is called as recessive one. So each one dominant and recessive is there. So let us see one by one. Case 1. First case, we can see what happens when we crossbreed these 2 plants. What is the condition? So in the first case, you can see it is called as monohybrid cross. 2 types of crosses are there. One is monohybrid and next one is dihybrid. First we are studying about monohybrid cross. That is a cross between 2 plants. Different. We are taking the character, only one character. In garden pea plants, 7 contrasting characters you have seen. But in the first experiment, we are doing a monohybrid cross, so that we are taking only one character. You can see that here we are these two plants are crossing. Which are the two characters we are taking? The nature of the flower. You can see that, the position of the flower. One is, you can see that with the terminal flowers, and the other one with axillary flowers. So, axillary flowers and terminal flowers. Axillary flower is the dominant character. Dominant, and recessive. Dominant character is axillary, and recessive is terminal. So, terminal flowers and axillary flowers. So, we are crossing these two plants. Let us see. Axillary with how we can show with the dominant capital A, capital A. Just we are taking any symbol you can take, because as it started with axillary A, that I took the symbol as AA. For the same terminal, we are taking small a, small a. Okay. When this capital A, that is dominant, with a recessive one, that is small a, small a. Here there is, what is the genes formed here, or the gametes formed here? This is in the F1 generation, the gametes formed only one type A, because no difference. A will be formed. See here one A, here one A. So there is no difference. I am showing only one A, as there is no difference. If difference is there, we have to show both it. Okay. So here also you can see that here with the terminal flowers A. Here also you can see another possibility of another one A, but of the same type. So I am not showing this one. So here this A combines with this A. What is the offsprings produced? With the one capital A and a small a. Got it? One capital A and a small a. So both the parents are homozygous with the capital A. With the small a, we have cross-pollinated these flowers, and you can see that the gametes formed here is capital A, here is small a, and here capital A, small a. And can you tell what is the nature of this flower? Whether it is axillary or whether it is terminal? Yes, as one capital A is present, this is the dominant gene, whereas this small a is a recessive gene. Got it? The capital A is the dominant one and the small a is the recessive one. So automatically this dominant gene, this is a superruling gene, this will suppress this character. Okay? This will not allow this character, that is terminal flowers, to show in this generation as it is present together. So automatically this plant will be axillary. Okay. Now come on, going to the next generation. With this plant, you can see that this produce what genes? This is the F1 generation or the first generation. This one is the F1 generation or the first generation. Going to the F2 generation, we are learning about F2, second generation. After one generation, next generation. Here you can see that parents plant capital A, small a into this. This is cross pollination we have done. Whereas here we are doing self-pollination with the same axillary plant having axillary flowers. Both the plants, but its genotype is different. Here it is pure homozygous genes are present, capital A with the capital A, and here small a with the small a. Whereas here when we capital A, small a, heterozygous, because this parent, you can see that we are self-pollinating these flowers with the capital A, small a. So here there is two possibility. What are the gametes formed? Capital A, yes, small a. Here you can see capital A, small a. So next generation, here there is no space, I will be showing here. You can see a checkerboard like this. Okay. Here first capital A, next one small a. So that here capital A, here small a. Now tell me, what is the genetic constitution of this one? I have shown you already in the case of hemophilia. I have shown you in the case of color blindness also. I have shown you. Come and tell me, what is the genotype? Yes, this one, A, capital A. Going to this one, this column, this capital A, small a. And coming to the third column, this capital A, this small a. And coming to the last column, this A and this A. Small a, small a. So this is called as F2 generation. That is the second generation. And here you can see, look into the genes of this one, capital A. So what is the type of this flower? Yes, this is axillary in nature. Coming to the second one, capital A, small a. Tell me. Yes, that is also axillary in nature. Third one, capital A, small a, axillary in nature. And fourth one, small a. Yes, see the grandparents, just like here it is small a. You can see that another one, terminal plant. Plant with the terminal flowers is forming in the next generation. Sometimes you will tell no, that when a child is you can see that a newborn baby is in the womb and they show the grandparents' character. You may wonder, it is nothing to worry. See, the same principle is applied there also. Here you can see the two generation, both the parents are, you can see here both the plants here are the plants carrying axillary flowers. But in the offspring, you can see in the 3:1 ratio. 3 axillary flowers, whereas 1 terminal flowers plant is produced. So in the second generation, this resembles to the first generation. Look. Actually, the parents both parental plants, they are not terminal one, both are axillary flower carrying one. But in the next generation, you can see that one plant is produced with terminal flowers. So this ratio, that is 3:1 ratio in the F2 generation, that is called as monohybrid ratio. A ratio produced when we are taking a single character, one character and crossing the plant. We are getting a 3:1 ratio, and that is called as monohybrid phenotypic ratio, means external character. What we are seeing externally. So monohybrid phenotypic ratio. Come on, another term is genotypic ratio, that is gene ratio. Here capital A, how many it is there? Only 1. Okay, only 1 is there. So see, here it is different, here it is different, here it is. So 1. Okay, then capital A, small a. Here 1 is there. Another one here. Any other? No. So 1:2, 2:2. Then small a, only 1. There is no other small a in the checkerboard. So here 1. So 1:2, 2:1 is the genotypic ratio, whereas 3 1 is the phenotypic ratio. Understood? The difference between phenotypic means externally. Here you can see externally we can say that these three plants, they are having axillary flowers, whereas the other plant, the last one, it is having terminal flowers. So this is externally visible. We can see it clearly. That's why it is phenotypic means it is visible. We can see. So 3:1 is the phenotypic ratio. But when we examine the type of genes in these plants, you can see that capital A homozygous dominant and 2 capital A small a, that is heterozygous, and another small a homozygous. This is the phenotypic ratio and this is the genotypic ratio. Thank you children. If you have